So this is just sort of a brief report on some of the stuff that we've done this year and talking about some of our plans for next year, I guess. So in 2025, we had 11 DNA days at Mount Rainier Nature Center. That is where we are meeting now and where we hold our DNA work. Um, you can see those are circled in red on the calendar. They sort of jumped around. In 2026, we are going to try to be more consistent and have the DNA days on the fourth Sunday of each month. So go ahead and mark your calendars. Make sure you don't have anything else scheduled on those days. We do have a little change in February that we already know about, and we may have to change a few others, but we're going to try to make it the fourth Sunday of every month so everyone knows when to come out and play with us with DNA. So what happens on a DNA day? There are sort of two ways the day can run, and some of it depends on the time of the year and what's happening out in the world. One option is to do what we call sample processing for eventual nanopore sequencing. And on those days, we take little bits of dried tissue from dried mushrooms that we've collected over time. They might be from Sequinota, they might be from various club members or other people who have dehydrated their mushrooms. We've got those all organized by genus. So we take the dried tissue, put them in tubes for 96 samples at a time. And then we go through a little bit of the process and send it off for nanopore sequencing to the Mycota lab. That means the results are delayed by weeks or months because it takes time to put together all of those batches, but we get a lot of samples processed and identified all at the same time. On some other days, we do the DNA extraction and PCR, or DNA amplification, for eventual Sanger sequencing all on the same day. So that's probably going to happen more often during the summer when we have a lot of fresh mushrooms out in the world and people are getting excited about them and bringing them in. And that means that we can process community samples on site. In those cases, we get the results in a week or so, and we don't process as many samples at a time. It's about 16 to 32, but you can watch the process. You can watch the DNA extraction, you can watch the PCR, and talk to us about everything that we're going to do with those mushrooms. So this is an image of option one. It's a great time. You can see all of the piled up bags of mushrooms that have been collected. We are organizing them, writing down the numbers, putting them on spreadsheets, double checking their identification on iNaturalist. And at the end of the day, we end up with batches of dried tissue in these little tubes that we hold on to for eventual nanopore sequencing. On the days where we actually do the DNA extraction and the PCR, we have sometimes just a couple of people. In one case, we had a number of students from Nature Forward. You can see in the right-hand picture that are watching me put together a gel. We do the DNA extraction, which is illustrated on the left-hand side. We take a small piece of fungal tissue, put it into an extraction solution and heat it, add a dilution solution, mix some of that with a mixture that will help make lots of copies of DNA, put it in a machine that will do that, and then see if we were successful. And you can see these bands here means that we did a pretty good job with those 8 samples. We then send it to a commercial company, in this case GeneWiz, to do this second part of the process, and what we get back are DNA sequences in the form of chromatograms. So just as an example of what we might get out of that sort of day, on November 16th, we did our 25th PCR, and that was with some of the students from Nature Forward. And here we've got the iNaturalist number, and we ask that everything that's submitted for DNA work has an iNaturalist number so that we have a unique number to follow it through. We have our field IDs. We're successful with 7 out of 8 samples, and this one was just Basidiomycota. So we have a nice band on that one, send it off for sequencing. We get back the chromatogram, we analyze that, and we end up with a series of letters, which are the nucleotides, which is our consensus sequence. We take that and put it into a public database that's run by NIH, and we do something called a BLAST search. That's basic local alignment sequence search. And that is one option. We can also do a BLAST with MycoMap, which is was developed by Stephen Russell, and MycoMap is just sequences of macrofungi and a few microfungi, but mostly just macrofungi. So whereas GenBank is everything, organisms from bacteria to viruses to fungi to animals, MycoMap is just mushrooms and has some sequences that aren't in the NCBI, the NIH database. If we're in the NIH database, we then get some results. So here's a list of our results. This was our basidiomycete. Turns out it's a Clitopolis. You have sort of your top hits, the things that are most identical or most similar. In this case, 99.82% similar. And that gives us some information. So we take that information and we add it to the iNaturalist observation. So in this case, this was our iNaturalist observation. It's got this one image of this nice little mushroom on some wood. The field ID was phylum Basidiomycota. We will add the DNA region that we have amplified. It's a barcode region. We'll put that in there and then put a little information about what we got out of that blast search at NIH. This isn't necessarily the final ID. iNaturalist is a conversation. People can suggest identifications and disagree with identifications, but we're doing this to put in more information. So the original iNaturalist observation had an image and a location. In this case, we're adding the DNA ITS barcode and some information from the blast search just to give people a guide as to what we saw. And then someone can come and say, hey, I want to know more. Use that sequence to compare to other sequences, talk to us about the sample. So it just really enriches the information we have for each of these observations. So in this case, out of our 8 observations, the Tricholoma imbricatoides was confirmed. The field ID Hygrophoracea was an Ampuloclitosobe clavipes. The basidiomycete was the Clitopolis. Unfortunately, the sequencing failed for one of them, which can happen. We had a beautiful band, but the sequences weren't good. The Rusupinatus is matching with something called Trichotis, but might not be actually that species. We had a Russula that's a Maryland specific Russula, or one that was first identified in Maryland, and the Calicinus citrina was confirmed. So in 2025, we processed 110 samples on those option 2 days where we did everything, and we've gotten 66 sequences uploaded to iNaturalist. We have a number more to upload from some of the more recent DNA days. And we also sent off this 2024 Sequenota samples to be sequenced via nanopore sequencing, and of those, 154 sequences have been added to iNaturalist observations. A few of those that were quite interesting, Karen Beal at Sequenota found a Pseudofistulina radicata. That was one of the Northeast Rare Fungi Challenge species. We now have it DNA confirmed. We've got a voucher specimen, and that one generated a bit of excitement because this population seems to be quite a bit more north than other populations that they knew about in Pennsylvania. You might also see some of the observations that you had updated with a new name, maybe a name you've never heard of before. In this case, what had been identified on iNaturalist as Cortinarius iodes came back as Phlegmasium calyptodermum. So Phlegmasium is a new genus that was erected to segregate out some Cortinarius. Cortinarius is a very, very big genus, and a lot of DNA work has been sort of sussing out who's where and how they fit together. And Phlegmasium, which is kind of not the most beautiful name, but is one of the names that came out. So if you look up this taxon, one of the things that it's easily confused with is Cortinarius iodes. They look pretty much the same. But we now have that much more information and records of Phlegmasium calyptodermum. And possibly because it's such a new name, but there just aren't that many observations out there of this fungus, and they pretty much need to be supported by DNA. But we now have added to the small number that are out there, and it just helps document the diversity and give us more of an idea of all of the fungi that are out and around us. This was also kind of an interesting story. In 2019, John Harper collected a Russula at Sequanota, and that was submitted to sort of the predecessor to our DNA committee, the Mycoflora Project. Um, and so that went into the Mycoflora Project and got sequenced. And those sequences are also in MycoMap and part of the MycoMap database. In 2024, a Russula was again collected at Sequanota. That was part of the most recent nanopore sequencing batch, and the ID on that came back as Russula Pennsylvania '08. And that means that's the eighth sort of unique Russula barcode to be identified from Pennsylvania. And if you compare to what's in the mycoblast database, there are 5 known accessions of this Russula. They're mostly from Pennsylvania, really from Sequanota, and there's one from North Carolina collected in 2023. So we're really, especially with the Sequanota observations, adding a lot of information to the diversity of some of these more cryptic taxa. So that's all I have to say. I'm happy to take any questions, and I want to thank MAWDC and PG Parks and really all the volunteers. We love having people come out and help and talk to people about what's going on. And thanks for listening. And I see a question from Chris Wozniak, and I will try to stop sharing my screen while I try to answer. All right. Tom, I don't know, should Chris put his question in the chat? Are we able to have people talk to us?
Oh, yeah. Let
Oh
me
yeah, thank you. Yeah,
Okay.
first of all, thank you. Nice explanation. I guess what I've been wondering for some time, and you answered it partially, is kind of like, what have we found? This is whatever, what, 3 years, 4 years, where we've been doing some DNA analysis, and I'm kind of curious, kind of where that has gone, and sort of what are the bona fides when you're done with that. We submitted a agaricus sample a few years ago, a recurring agaric in our woodlot that looks like, yeah, you should be able to eat this. It smells good. It looks good. It's got pink gills. But it's not anywhere in Kerrigan's books or other books, and it makes me nervous. And so we submitted. We did not get any response to it. But what I'm wondering is, I talked to Tom about this a couple of years ago. To me, it raises the question, what is a species? And as a biologist, I know that's not a simple question, whether you're talking about bacteria, fungi, viruses, mammals, whatever. And I'll just, again, Tom, reiterate, at some point, not that I have all the answers, but I'm willing to start the discussion. And my good friend John Cow, I know, will join me. If we want to discuss kind of what are the boundaries of a species and what does it mean to call something, agaricus X, whatever it is. And we have plenty of examples where things are just really kind of mushy. There are species complex where you might have a dozen species all called the same thing. I'm embarrassed to say that, 35 years ago, what I was eating and calling Armillaria malea is not Armillaria malea. I didn't die, but it's like, wow, you think you understand the boundaries of a species, but it's not necessarily simple. So just putting it out there that, I think given the context of this DNA database, that a basic question is what does it mean if you identify something, whether it's through the NIH thing, which again, John Cow and I have contributed sequences for bacterial proteins and things, and arbitrarily came up with a cutoff, 95%, that's the same species. Well, that's just a guess. We don't have the answers kind of thing. But I think it's important that people understand that because, whether you're talking about the Endangered Species Act, whether you're talking about just common knowledge, I don't think a lot of people really think about what does it mean when something is a species designation. So I'll shut up now. Sorry.
Oh, no, and thank you for that. And that's, in fungi, of course, until very, very recently, the species concept was a morphological species concept. So we don't even have a biological species concept in fungi. It's if it looks like this, then it's this. And so, whereas with birds or, mammals, you can have a biological species
Right.
concept. Even with plants, you can have a biological species concept. With fungi, it's what did they look like? And of course, nowadays, almost everything is based on a phylogenetic species concept, which, as you were alluding to, is somewhat arbitrary. And morphology is somewhat arbitrary as well. Um, it's just kind of who cares about it, and so, people have kind of left a cutoff of 97%, but that might depend on what family you're talking about and what genes you're talking about and what gene regions you're looking at and how quickly they evolve and all of that. So, I mean, my kind of answer for people is I think it depends on why you want to know a species. Like, are you looking to see whether it's something you can eat? Is it as closely associated with something that visually looks like something you can eat and you don't want to kill yourself? Or is it a more academic question of how many species, however we define them, are in this particular
Yeah.
area?
And in some cases, maybe, do I really need to know?
Right. Yeah. Yeah. And
I'd like
so,
to know, but do I
yeah.
need to know?
And right now, so in terms of outputs, there aren't that many of us doing this. And we are right now we've gathered a lot of data. So we have a lot of sequences. And like I said, we're trying to put them on iNaturalist so they're out there for anybody to use who's interested in them. If there's a particular taxon they're interested in, we want our sequences out there. We'd like to do some of the work ourselves too, but I personally haven't had the time to go through and do phylogenetic analyses on everything that we've generated. So we've been kind of focusing on just sequencing and getting the sequences out there, um, and having as much coverage as we can,
Yeah.
and hoping in the next few years to have more time to actually delve into some of the taxa that we're more interested in.
And we appreciate the work you guys have done. I know it's a lot of work.
Yeah, no,
So
it's
thank
it's
you.
it's a lot of fun too. So come volunteer.